Hey everyone! Hope you're all doing alright today. Let's get into O-level biology. Lesson 5 is enzymes and rates of reaction. Enzymes are proteins that act as biological catalysts, increasing reaction rate without being used up. A substrate with a complementary shape binds at the enzyme's active site, forming an enzyme-substrate complex. Products then leave and the active site can work again. Enzymes provide an alternative pathway with lower activation energy. Specificity means that one enzyme usually catalyzes one reaction or a narrow group. Amylase hydrolyses starch, protease hydrolyses protein, and lipase hydrolyses lipids, but these names describe substrates rather than where enzymes act. At low temperature, particles move slowly, so successful collisions between substrate and active site are infrequent. Warming increases kinetic energy, collision frequency, and reaction rate up to an optimum. Above the optimum, vibrations disrupt bonds, maintaining the enzyme's three-dimensional shape. The active site changes and substrate binds less often. This denaturation may be irreversible. Cooling does not normally denature an enzyme, so activity can recover on warming. A temperature rate graph therefore rises gradually and falls steeply after its optimum. pH changes charges and bonding within a protein, altering active site shape. Each enzyme has an optimum pH related to its working environment. Pepsin functions in acidic stomach conditions, whereas many intestinal enzymes work best in neutral or alkaline conditions. Far from the optimum, activity falls and denaturation may occur. Enzyme concentration raises initial rate if substrate is abundant because more active sites are available. Increasing substrate concentration raises rate until all active sites are occupied. The graph then plateaus because enzyme concentration is limiting. To investigate amylase, place iodine drops in a spotting tile. Equilibrate amylase, starch and buffer at a chosen temperature, mix them and transfer samples to iodine at fixed intervals. The endpoint is when iodine remains yellow-brown, suggesting no starch is detected. Repeat at several temperatures while controlling pH, concentrations, and volumes. Rate can be estimated as one time. A shorter endpoint time means a faster reaction. Sampling interval limits precision, so continuous calorimetry can provide more detailed data. Immobilized enzymes are trapped in beads or attached to a support. Substrate flows over them and products are collected without enzyme contamination. The enzyme can be reused and may be more stable, reducing cost, although diffusion into beads can slow reaction. Lactase can produce lactose-reduced milk, while pectinase clarifies fruit juice. When evaluating industrial conditions, balance reaction speed against denaturation energy costs and contamination risk. The highest temperature is rarely the best operating choice. Calculate rate as amount changed time, or use one endpoint time when the same endpoint is used. Describe a graph by trend, optimum and decline, then explain each region mechanistically. Starch disappears in 100s at 20 degC and 40s at 35 degC. Relative rates are 0.010s1 and 0.025s1, so the reaction is 2.5 times faster at 35 degC under controlled conditions. Treat each checklist item as a gate. Skipping labels or units is how marks disappear even when the idea was right. Do not say enzymes are killed, that heat always increases rate, or that substrate concentration denatures enzyme. Distinguish endpoint time from rate. Practice. Explain the steep fall in activity above an enzyme's optimum temperature. Answer: heat disrupts bonds, maintaining tertiary shape, changing the active site. Fewer substrates bind and fewer enzyme-substrate complexes form. Watch the numbers land in the matching symbols. Check that units cancel cleanly. If they do not, the setup is wrong even before the arithmetic. Practice. Endpoint times are 80s and 50s. Calculate both relative rates and the rate ratio. Answer. Rates are 0.0125s1 and 0.020s1. The second is 1.6 times the first. Watch the numbers land in the matching symbols. Check that units cancel cleanly. If they do not, the setup is wrong even before the arithmetic. We'll leave it there for enzymes and rates of reaction. When we come back, it's plant nutrition and photosynthesis. Hope that helped. Catch you next time.